In this lecture, we'll be discussing recovery of function. We'll be discussing what we mean by recovery of function, and we'll look at research that informs our understanding of this notion. Much of the material that's covered in this lecture can be found in Chapter 2 and Chapter 4 of the Shumway, Cook and Woolacott textbook. So, what do we mean by recovery of function? Let's start off by just focusing on the word recovery. A stringent definition of recovery can be stated in the following way. A recovery has occurred when functional goals can be achieved in the same way that they were pre-injury. In this definition, when we're talking about in the same way, we're talking about using the same processes in the same way that they were used prior to the injury. We can also have a less stringent definition of recovery. For example, recovery can be defined as the ability to achieve task goals using effective and efficient means, but not necessarily those means that were used before the injury. Now let's turn to the challenge of defining what we mean by function. Here we have a definition by Craik. For Craik, function refers to the complex activity of the whole organism that is directed at performing a behavioural task. For Shumway, Cook and Woolacott, the authors of the textbook, when they're talking about function, they're talking about skilled action. So here we're thinking about function in terms of complex goal-directed activity in our first definition and skilled action in our second definition. The difference between the stringent and the less stringent definitions of recovery motivate us to think about a distinction between what we might call recovery on one hand and what we might call compensation on the other. In the context of thinking about function in terms of skilled actions, motor recovery refers to the reappearance of a specific motor pattern that was present prior to the central nervous system injury. In contrast, motor compensation refers to the appearance of new motor patterns. These motor patterns can result from either adaptation or substitution. In the case of adaptation, we're talking about the remaining or intact motor elements being reorganized in some way so as to preserve the function. In the case of substitution, functions are taken over, replaced, or substituted by different end effectors or body segments. In both cases, we see that function is returned, but it's not returning in its pre-morbid form. So where should therapy be directed? Should it be directed at recovery of function, or should it be directed at compensation? Historically, researchers supported the idea that the adult mammalian central nervous system was rigid and unalterable, that function was believed to be localized in various parts of the central nervous system, and that regeneration and reorganization were not possible within the adult CNS. These results have historically led to the conclusion that therapy should be directed at compensation. Modern neuroscience has revealed a less bleak picture, with a solid body of research supporting the conclusion that the central nervous system has great plasticity and a great capacity for reorganization. This research supports a capacity for reorganization and a potential for recovery of function, as well as compensation. There are many factors that are known to affect recovery of function. On this slide, we have a summary of the known factors. After we've reviewed this summary, we'll focus in on the evidence that's led to some of the most notable factors to be identified. The factors affecting recovery of function can be classified into factors within the individual and factors within the environment. Factors within the individual, or endogenous factors, include a patient's weight, gender, brain trophic factors, genetic factors, lesion factors, age, and premorbid factors. Factors within the environment are factors that are external to the individual, or exogenous factors. These include pre-injury factors, like a patient's pre-injury dietary restrictions, a patient's pre-injury exercise, and a patient's pre-injury level of environmental enrichment. Each of these provide examples of neuroprotective factors. Post-injury, factors within the environment that affect recovery of function include pharmacologic functions and rehabilitative treatments. So let's start reviewing the evidence that shows that certain factors are implicated in recovery of function. We'll start by looking at age. Recovery of function is affected by the age of the individual at the time of the lesion. Kennard examined the functional consequences of surgically removing the motor cortex. He observed marked differences as a function of whether the procedure was performed on adult monkeys or infant monkeys. Kennard discovered that following the procedure, infants were able to relearn how to feed, climb, walk and grasp objects, whereas adult monkeys were not. In another study, Anderson and colleagues studied this effect in human language function. They observed that damage to the dominant hemisphere showed little or no effect on speech in infants, but caused various degrees of aphasia in adults. The take-home message of this research is that the same brain regions will show different capacities for regeneration at different ages. The effect of age on recovery of function is not straightforward. Injury to some areas of the brain can lead to similar deficits in both infants and adults, and damage to other brain areas can show little effects in infancy, but problems can develop later in life. One specific example of this is injuries to the frontal cortex. So, what's the logic that allows us to understand why the effect of age on recovery of function is not straightforward? If the area that's injured is mature, then injury will cause similar damage in infants and adults. If, in contrast, there's an immature area that's functionally related to the injured area, then that area may assume the function of the injured area. If an immature area is damaged, and no other areas assume its function, then no problems may be seen in infancy, but in later years deficits may become apparent. In summary, data on age-related effects on brain injury suggests that the brain reacts differently to injury at different stages of development. Now let's look at how the characteristics of the lesion affect recovery of function. Held observed that the extent of recovery depends upon the characteristics of the lesion. For example, if a lesion is small in size, then there's a greater chance of recovery, as long as no functional area has been completely removed. Moreover, if lesions develop slowly, then there's likely to be less functional loss. The effect of lesion characteristics was investigated by Travis and Woolsey. In this research, they caused lesions in the motor cortex of monkeys. They investigated the consequences of producing serial lesions in the test animals, and they manipulated the amount of time that the animal had to recover between lesions. The lesions were produced in Brodmann's area 4 and 6 in one of two ways. Either a single lesion was produced at one time, or a lesion of similar size was produced serially over time. In the case of the single lesion, test animals became immobile. In contrast, when a similar lesion was produced serially over time, allowing time for the animal to recover between lesions, then function was spared. We've just looked at factors of age and lesion characteristics. They were both examples of factors within the individual that affect recovery of function. Let's now switch to factors within the environment, and let's start by looking at the neuroprotective effects of experience. As we'll see over the next few slides, recovery of function is affected by the experience of living in a particular environment, and the experience in engaging in particular routines and types of activity. This effect holds both when the experience has occurred before injury and when the experience occurs after an injury. Some valuable context for understanding the impact of experience is research that investigates the effect of raising rats in impoverished versus enriched environments. Rats raised in enriched environments show increased brain weight, increased dendritic branching, and increased enzyme activity. Research on this topic has shown that there are wide-ranging benefits of early exposure to environmental enrichment. For example, environmental enrichment can prevent drug addiction. A study in 2009 demonstrated that mice exposed to environmental enrichment were less affected by the consequences of cocaine exposure in comparison to those in a standard environment. Let's focus in on some specific research concerning pre-injury experience. Pre-injury experience of an enriched environment has been shown to protect animals against functional deficits that result from brain lesions. Held studied the task of rats learning mazes. In this research, cortical lesions were given to a group of rats that did not have preoperative enrichment. This was the control group, and a group of rats that did experience preoperative enrichment. In this research, the enriched environment included ramps, elevated platforms, a swing, climbing objects, wooden corks, and ping pong balls. So, what were the findings from this research? After the surgery, the enriched animals made fewer mistakes in maze running compared to the control group rats. The post-surgery rats from the enriched environment performed so well that they actually outperformed the abilities possessed by the control group rats before they were lesioned. Now let's turn to post-injury experience. Stein and colleagues studied rats with unilateral lesions of the visual cortex. In order to study the effect of post-injury experience, rats were divided into two groups. During recovery, the rats in Group 1 were allowed to move freely in the environment, and they were allowed to interact with the visual cues. In contrast, during recovery, the rats in Group 2 were also exposed to visual cues in their environment, but they were restrained from moving. The results of this research are compelling. The rats that were allowed to move freely in their environment experienced a good recovery of visual function. In contrast, the rats that were restrained from moving were left with severely impaired visual function. So what are the clinical implications of these results? What we're learning here is that if environmental stimulation is to affect recovery of function, it must incorporate active participation of the patient. Without active participation, a full recovery will not occur. In a second study investigating the effect of post-injury experience, Ogden and Franz caused hemiplegia in monkeys by creating motor cortex lesions. In this research, the lesioned monkeys were placed into one of 4 different groups that varied in the type of post-operative training that was received. The first group of lesioned monkeys received no post-operative treatment. The second group of lesioned monkeys received general massage of the involved arm. A third group received a treatment that involved the non-involved limb being restrained. And the last group was given a treatment that included restraint of the non-involved limb, stimulation of the involved limb to move, and forced active movement of the animal. 3 weeks after the lesioning operations, the only group to show improvement was the last one. The takeaway here is that recovery benefits from setting up a training environment that puts the effective limb into active and functional movement. We just looked at research that explores how type of training affects the recovery of function. Now let's look at a study by Black and colleagues that investigated both type of training and schedule of training as potential factors. Black and colleagues caused lesions in the forelimb area of the motor cortex of monkeys. They studied the recovery of function after 6 months of training. By assigning the lesioned monkeys to different groups, they investigated how differences in when training was initiated, as well as the focus of training, upon the recovery of function that was observed. To study the impact of time of training initiation, they either had training begin immediately after surgery or after 4 months. When recovery of function was measured at 6 months, they observed much better recovery of function if training had been initiated immediately after surgery. To study how changes in the focus of training impacted the recovery of function, Black and colleagues compared the effectiveness of training protocols where the focus was either solely upon the normal or uninvolved limb, or was focused upon the involved hand and the normal hand together, or was focused solely upon the involved limb. As you might intuit, only training the spared limb was minimally effective. Training the involved and spared limbs together was much more effective, and focusing training efforts on the involved limb was the most effective. We've just been looking at investigations into how the specifics of post-injury experience impacts recovery of function. The studies we've looked at so far present a seemingly very simple and clear picture of the types of post-injury experience that we might consider to be best. When interpreting such findings, it's very important to bear in mind that the effect of post-injury rehabilitation training, both upon neuroplasticity and upon recovery of function, is complex and is affected by a series of factors. For example, early and intense intervention is not always best. Early and intense motor enrichment can promote neuroplasticity in the contralesional hemisphere, but it can also exaggerate the effects of injury in the perilesional area. Work on the topic by Shalit and colleagues has shown that neuroplasticity and recovery of function in perilesional areas is facilitated best by gradual and modest increases in motor therapy. So far, we've taken a quick look at pre-injury experience and a quick look at post-injury experience. How do the benefits of pre-injury versus post-injury experience stack up against each other? To examine this question, Held looked at the potential for recovery of function in rats after their sensory motor cortex had been removed. Held assessed function by looking at the ability of the rats in the study to locomote across a narrow track. The rats in this research were assigned to either a sham lesion group or a lesion group. The sham lesion group still experienced the negative effects of an operation, but they did not have their sensory motor cortex removed during the operation. These two groups of rats are then further assigned to one of 3 enrichment groups. The different groups of rats either experienced no enrichment, either before the operation or after the operation, enrichment before the operation, or enrichment after the operation. In this research, the enriched environment was a large wire mesh cage that had dimensions of approximately 1.5 meters by 1.5 meters by about 50 centimeters tall. In this cage, there were elevated platforms along the 2 sides of the cage. There was a wide ramp that ran from the floor to the lower platform, and a second wide ramp with circular cutouts connected to the lower platform to the higher platform. So, what were the results of this study? For the rats in the lesion group, the rats that experienced the enriched environment after the operation recovered more quickly than the control group that experienced no enrichment. It's important to note that these post-operatively enriched rats never regained full locomotor functions. When we look at the recovery of function in the rats that experienced an enriched environment post-operatively, we see that they recovered a level of function that was equal to that of the rats in the sham lesioned and enriched control groups. This conclusion was based upon both behavioural measures, such as measures of track running time, as well as more fine-grained movements analysis, such as by looking at the variability of their gait. So what are the takeaways from this research? The main conclusion to draw is that post-operative enrichment is effective, but it's not as effective as pre-operative enrichment. We can interpret the findings of this research in the following way. Enriched subjects may have neural circuitry that is more functional, that's more varied or more degenerate, to use a term that we used earlier in the semester, than that of comparatively restricted subjects. By restricted subjects, we mean the subjects that were in a less enriched environment. This more functional neural circuitry may provide an increased ability to reorganize the nervous system after a lesion, and to make use of alternative neural pathways to perform a task. We'll be exploring the idea of alternative neural pathways quite soon. In the final part of this lecture, I want to talk about some of the practical applications of motor learning research. More specifically, what are some of the takeaways from the study of motor learning that have implications for retraining patients? Let's start by looking at the application of a very simple concept from motor learning theory, the concept of knowledge of results. Knowledge of results, or KR as we may write it on this slide, involves the giving of information about the outcome of a movement. We're going to be looking at the application of knowledge of results as it relates to treating Parkinson's disease. Parkinson's disease is associated with compromised proprioceptive feedback, difficulty integrating sensory and motor information, and difficulties with the timing of the control of actions. Guadagnoli and colleagues investigated the hypothesis that patients affected by Parkinson's disease may benefit from feedback in the form of knowledge of results. They investigated a task in which participants used a stylus on a digital drawing tablet to produce a movement path from a starting location to a goal. Here we see one version of the task. Participants start with their stylus positioned in the circle at the bottom, and they then move to the goal, while making sure to avoid contact with the barrier. Participants were tasked with performing this task in a specific way. They had to make the movement in a specific amount of time. The goal then here is not just to get to the goal, but to get to the goal at the right moment in time. The goal time was set to 65% of a participant's fastest task completion time. Now that we've set this up, we can finally get to talking about providing knowledge of results. After each trial, participants were given feedback on their movement time. For example, they might be told that their movement was 20% too fast. In this research study, participants were either patients affected by Parkinson's disease, or they were age-matched controls. Participants from each group were split into two groups. Either they were assigned to a group in which they received feedback on movement time on 20% of trials, or they were assigned to a group when they received feedback on their movement time 100% of the time. What we're interested in here is the question of how often should knowledge of results feedback be provided in order to promote the best training. In this experiment, participants completed an extensive practice in which they received knowledge of results. After the practice, they performed a retention test, where the researchers assessed participants' ability to retain the skill that had been trained. Here we see the results. On the y-axis, we have timing errors. In this graph, a value of 0 means that you are arriving at the goal at the trained time. Values above 0 mean that you have either arrived at the goal before or after the trained time. On the left side, we have the results for the age-matched control, and on the right we have the results for the Parkinson's disease group. The white bars are for the participants that received feedback on 20% of the trials during training, and the grey bars are for the participants that received knowledge of results feedback after every single one of the trials. When we look at the results for the age-matched controls on the right, we see that they performed the task better in the retention test if they received less frequent feedback. For the age-matched controls, we see that giving feedback after every single trial actually hinders learning and subsequent performance in the retention task. There are many reasons for this result, but one reason is that when knowledge of results feedback is too frequent, learners can become too reliant on the information that's provided and can neglect the processing of intrinsic feedback that they will have to rely on when the additional knowledge of results feedback is no longer available to them. On the right-hand side of the graph is the results for the Parkinson's patient group. We see the opposite result here. Unlike the age-matched control group, here we see that giving feedback after every single trial actually improved learning and subsequent performance in the retention. So what are the takeaway messages here? The general takeaway message is that principles of learning that are derived from the study of healthy adults and athletes don't necessarily apply to the same way to people with pathologies. The more specific takeaway message here is that Parkinson's patients are better able to make use of the knowledge of results that was provided during practice. The authors interpret this finding in the following way. They propose that knowledge of results is providing an external source of information. For the Parkinson's patients, the feedback in the experiment is providing a source of information that they would not otherwise be able to interpret through their own feedback system, and that they would not otherwise be able to integrate into their own actions. The next concept from motor learning theory that I want to look at is mental practice. Mental practice refers to mentally rehearsing an action. The benefit of this type of practice is that it can be done by a patient without risk of injury, since they're not actually performing the action. In the literature, the benefits of mental practice have been demonstrated in multiple areas, including the rehab of injured athletes, elderly individuals with balance problems, and post-stroke patients. Let's look at some evidence that suggests that mental practice can play a role in the recovery of function. Here we're going to be looking at some research performed by Mowlin and colleagues that investigated the possible benefits of mental practice in the rehabilitation of patients recovering from a stroke. The focus of the research was on training greater use of the affected limb during sit-to-stand transitions. In this particular research study, sit-to-stand transitions had to be performed without using the hands. So only subjects that could stand up and sit down without assistance from their hands were recruited. The desired outcome of this intervention study was to increase the loading on the affected leg while participants stood up and sat down. All participants in this study completed a series of tests designed to assess their working memory capacity. This was done because the effectiveness of mental practice is known to depend upon a person's imagery ability. For example, some people have great difficulty imagining a described action, whereas other people can imagine an action with a high degree of vividness and control. To test working memory, the experimenter presents a series of items, which the subject has to reproduce immediately in the remembered order. This includes remembering 2 to 3 commonly used words. This is a test of verbal working memory. Tapping on blocks in a particular order. This is a test of visuomotor working memory. And producing a series of gestures in a particular order. This is a test of kinesthetic working memory. Before we can get to the results of this study, we need to understand how participants were trained to perform mental practice. The mental practice training protocol in this study had participants performing sit-to-stand and stand-to-sit transitions in an apparatus where they had their feet on force platforms and they had a display screen in front of them. The force plates measured how much each limb was being loaded during the transitions by integrating, that is, adding up the force over time that was exerted by the limb. So here we see the ground reaction force over time, and what we do is we add up the amount of force over time, and we're going to give that as feedback to the participants. More specifically, after each time that the participant has sat down and stood up, they're given information about the degree to which they were overloading the limb on their unaffected side. Using this feedback, participants were instructed to modify how they planned and executed the task in order to reduce the overloading on the unaffected limb, while also increasing the loading on the affected limb. As they did this, they were also asked to relate the strategy they were trying to the outcome viewed on the screen. They were asked to try and remember the feeling and the movement sequences that were associated with success or error, in order to develop an inner image of their performance. Participants were also instructed to verbally describe the things that they did that improved their performance, with the goal of using these descriptions as pointers later during mental practice. For example, a participant might describe that they shifted their body to the right and then moved forwards and up, and then recall this later during mental practice. Now that we understand the tests of working memory that were used and the training protocol that was used to get participants to be able to mentally practice the activity, let's actually go and look at the specific training protocol. For the training protocol, participants repeatedly performed blocks of training trials. Within each block, there was 1 trial of physical practice and 5 trials of mental practice. In the physical practice, patients stood up and then sat down when they heard an auditory cue. For the mental rehearsal, they were instructed to close their eyes and to imagine that they were standing up and sitting down, and to signal verbally at the beginning and end of each repetition of mental practice. The degree to which participants were overloading their unaffected limb was assessed before training, after the training session, and 1 day later. We can see in the graph that the largest degree of overloading was in the baseline measure that was taken before training. When we statistically compare the post-training measurement and the follow-up measurement to the baseline, we can see that there was a reduction in the degree of overloading in both cases. So, what are the main results of this study? Participants showed an increase in the loading of the affected limb during transitions between sit and stand. Improvements were maintained on a retention test 1 day later. Now, we're going to get to the most interesting part. The greatest improvements on reducing overloading the unaffected limb were shown by the patients that had the highest visuomotor working memory scores. Here we see a graph where the y-axis is the participant's visuomotor working memory score, and the x-axis is the amount of improvement in reducing overloading on the unaffected limb. We can clearly see that those patients that have high visuomotor working memory scores also show the greatest degree of improvement. When we specifically focus on those patients that have impairments in working memory, we see that only a small improvement resulted from training, and that there was no retention and follow-up. In sum, we're seeing that mental practice can be useful, but that its usefulness depends upon the specific cognitive resources available to the individual patient. In this lecture, we introduced the topic of recovery of function and looked at some of the foundational research on this topic. We also looked at some of the practical applications of motor learning research as it pertains to the treating of patients.